So that's why we saw three tall plants and one short plant. So similar way we do traditional plant breeding in rice also. Okay. So this is also a self-pollinating crop. Rice is a self-pollinating crop. That means it has both stamen and stigma within it. So how we do traditional plant breeding in rice? Suppose there is a one plant which is susceptible for a disease. You can see this leaf. This is a bacterial leaf-like disease symptom. This I'll tell you later. So this plant one is susceptible for bacterial blight disease. Okay. Now we have another plant, another rice plant, which is say resistant for the disease. There is no disease symptom here. So now we make cross between these two plants. We get hybrid, and when we self-fertilize this hybrid plant, we get thousands of F2 plants, thousands of plants. And these plants now we can screen for bacterial blight disease, and whichever plant will show the disease resistant, that plant we can select and we can use as a disease resistant plant. This is a traditional plant breeding. Okay. So how we do crossing in rice? If you see this rice spikelet, it has 6 anthers, okay, which has pollens, and then it has 2 stigma. Okay, so to do or to make crossing in rice from the recipient plant, what we do, we emasculate the anthers. We remove all these anthers, so it will have only stigma, and then from the donor plant with that has this desired rate, we then bring these pollen dust. Okay, we do pollen dusting. We bring uh, the stamen and do dusting here. So in that way, it gets fertilized, and we get a hybrid plant. Now coming to marker assisted breeding. Okay, so this is a molecular using the molecular biology tool. As I mentioned earlier, for each trait there are two alleles, okay, and we can differentiate alleles. If both the alleles are same, then it is called as homozygous condition. And if both the alleles have a different size or different there is size in difference, then it is called as heterozygous condition. You can see here maybe two bands, right? So each one is one allele. Okay, that gene has two different alleles. You can see two bands, that means it is heterozygous condition. Here you can see only lower band or one kind of band, like in this case only upper band. So this is called as homozygous condition. Okay, so suppose for disease susceptibility or for bacterial blight disease, the allele size is bigger one. The upper one is for susceptibility, okay, and the lower size is for resistant. In this plant is resistant for bacterial blight, and we see this lower size. When we make cross, we get hybrid plant. Then we self-fertilize, we get thousands of then F2 plants. In this case, in marker assisted selection, we don't do phenotypic selection. What we do, we look for these two alleles. Okay. What we want from here is we want only this allele because it is coming from resistant plant, this lower size band. So then we check for these alleles, and we select only those plants which have that resistant allele, that lower size of allele. You can see in these arrow marks. Okay, wherever there are only that lower band, only those plants then we select, all other plants we discard. So in this case, we handle only few plants that is of our interest. So we can reduce lot of our work through this method. So with this background, now I'll speak about the disease. So bacterial blight is a serious disease of rice. Okay, and this is the bacteria that cause the disease. The bacteria is called as Xanthomonas coryzi parvovirus. Okay, and this is how it looks like on a plate. It looks yellowish in color because it produces a yellow pigment called xanthomonidin. Because of that, the color of bacterial colony is yellow. When we inoculate a rice leaf with this bacteria, okay, you can see two leaves here. One leaf here, this leaf is inoculated with just water. So how we inoculate? We grow the bacteria in a liquid culture. We dip a Caesar, okay, normal Caesar, or and then we make a cut on the rice leaf. And after 10 to 14 days, then we can see the disease symptom. You can see yellowing in the rice leaf. Okay, this is a disease symptom of bacterial leaf blight disease. This leaf was cut with the Caesar that was dipped in just normal water. So there is no disease symptom, and this is bacterial leaf blight disease symptom. Okay, and in severe condition, in the field condition, what happens? All the plants, the whole field looks like dried out. So that means all the plants are dead, and there will not be any crop yield. So that will be a loss for the farmer because he will not get any crop yield. And what we have seen is there are no effective controls to control this disease. Okay, I'll mention what are the difficulties. So in this interaction, we have studied in more detail, like how this bacteria cause disease in rice plant and how rice plant try to defend itself. So there are four level of interactions. Okay, so this is a single bacteria. This is electron microscopy image of a bacteria of Xanthomonas. And what it does, it secretes cell wall degrading enzyme, okay, that are virulence factor or that degrade rice cell wall, and then that degradation, that plant detects and induce defense responses against the bacteria. Okay, and bacteria is again smart, so what it does, it suppress those defense responses. Okay, and if it is able to effectively suppress defense responses, that means again it is able to cause disease. But plant has another level of defense that is mediated by resistance genes. Okay, it can it can recognize these molecules which suppress defense response, and it can block the action of those molecules, and that's how then we get a defense response, and there will not be any disease. So like that, there are four level of interaction. Okay, let's see here. So how this bacteria cause disease? First step is attachment. Okay, it reaches to rice leaf. Then once it gets attached, it has to go inside. So then it moves through. You can see flagella here. It will also have pili, small pili. It will go inside. Once it go inside, what is difference between plants and animal cell? Plant cell has this thick cell wall. Okay, so this is a thick barrier for the bacteria. What bacteria wants from a plant? It wants nutrition. And where is nutrition in the cell? It is inside the cell. So to you can see this whole green, uh, this portion is a single plant cell. Okay, this is one part of the leaf. Uh, So, in order to get nutrition from a cell, the bacteria has to degrade this plant cell wall, which is the outer layer of the cell. Okay. So, what bacteria then do? It it secretes several cell wall degrading enzymes. Cell wall degrading enzymes. Okay, like cellulase, xylanase, pectinase, and then those enzymes degrade the component of plant cell wall, so there will be breakage in the cell wall. But then plant is also smart. It will detect those degradation product that something has attacked uh, that cell, okay, plant cell. So I have to do some defense response. So what then plant cell will do? It will induce defense related genes, okay. Ultimately it will induce a level of phytohormones or antimicrobial compounds, secondary metabolites. So in that way it will try to defend itself from the attack of the bacteria. So then, to suppress this defense response, bacteria secrete another set of molecules called effector molecules, okay, through another injection like machinery. So these molecules are secreted directly inside the cell, okay, and then these molecules suppress the defense response induced by plant. So if suppression is effective, the bacteria will be able to cause disease. But then in the plant cell, there will be fourth layer of defense that are resistance genes. Okay, these are very specialized genes, and they can block the effectors molecule, action of effector molecules. In this way, there is fighting between bacteria and plant cell, and whoever wins in this fight, if bacteria wins, there will be disease on the plant. If wins, then the plant will be resistant for the disease. And what kind of defense responses we see on rice? So one is, this is called callose deposition. You can see this white dot here. So actually, this is like when we get any cut on our skin, we see blood clotting, right? So callose deposition is also like that. Whenever there is attack of a say insect or bacteria, we can see these white spots. So they try to strengthen the cell wall. They try to block the spread of the pathogen. Then we see hypersensitive response. You can see this uh, yellowing region. So in this case, in rice leaf, if we infiltrate bacteria, that is uh, uh, through syringe, 1 ml syringe without needle, okay. If plant try to restrict the pathogen, then what it does, it try to kill only this portion so that bacteria should not migrate, should not move further. Then also what we have observed are changes in level of defense hormones. Plant has jasmonic acid, salicylic acid. These are defense hormones. Also changes in level of secondary metabolites, changes in expression of defense associated genes. So these kind of different defense responses we observe on rice plant upon pathogen attack. So now coming to the story of improved Samba Masuri rice, okay, how we develop. So as you all know, rice is a staple food of India, and in terms of area and production both, this is one of the most important crop in our country. And Samba Masuri is one of the popular rice. This Samba Masuri or Sona Masuri both are same. You can see in supermarket or maybe what rice you eat generally. It is Samba Masuri here, particularly in Telangana. So it is one of the very popular rice variety which is cultivated in about 10% of total rice cultivation area. It has exceptional quality, and also it is high yielding rice variety. But this variety is susceptible to many diseases, including bacterial plague. Okay, and what we have observed, our farmers also came to us and they told that there are no effective chemicals to control the disease, and the best way to control this disease is to develop resistant rice varieties. Okay, and there are multiple resistance genes for uh, bacterial blight disease resistance. And if we transfer just one gene, maybe it will be resistant for one uh, one strain of the pathogen. Also, you should notice that there are hundreds of different strains of pathogen. like what we see now in the case of COVID. Uh, First, we saw the first phase, then Delta came, then Omicron came. So, like that, we are seeing different version of this COVID virus. So, like that, this bacteria Xanthomonas also has different strains. Okay, and if we put just one gene, then it may not be effective. So we need to put multiple genes, multiple resistance genes to get a better resistance. So then what we did, so this is a Samba Masuri rice, okay, which has fine quality, which is a high yielding, but it is susceptible to bacterial blight disease. There was another rice variety which has a poor quality, but it was having three bacterial blight resistance genes, which are called as XA21, XA13, and XA5. So, like I mentioned, the marker assisted selection, we used that strategy, and then we brought these three resistance genes into Samba Masuri. Okay, we made process between Samba Masuri and this variety, and then we select for plants that has these three resistance genes and also the quality like Samba Masuri. So that finally the developed variety we called as improved Samba Masuri rice. So this rice has all the good quality of Samba Masuri. It is high yielding, but then it is resistant for bacterial blight disease because it has these three resistance genes. Okay, and this variety is now already in farmers field. So now you can see this leaf A is Samba Masuri leaf. When we inoculate with bacteria, with Xanthomonas bacteria, and after 14 days, we can see a disease symptom. And this leaf B is improved Samba Masuri leaf. Similar way, when we inoculate, after 14 days, we wait for 14 days, but there is no disease symptom. That means because of those three resistance genes, this plant is now resistant for bacterial blight disease. Okay, and this is in real field situation, field condition. You can see this field is Samba Masuri field, and you can see it is severely affected by bacterial blight disease. And this field is improved Samba Masuri rice, and it is free from bacterial blight disease. Also, quality wise, grain looks similar. This is Samba Masuri, this is improved Samba Masuri. Also, after cooking, also it looks similar. And to popularize this variety, we have so far distributed seeds to about 10,000 farmers in different states like Telangana, Andhra Pradesh, Tamil Nadu, Karnataka, Chhattisgarh, Jharkhand, Uttar Pradesh, and few other states. This is a representative picture of farmers there in Tamil Nadu. So he's actually Dr. Ramesh Sonti who developed this variety. I was working with him, and now I'm taking care of this project. I'm continuing the work. And this variety was developed in collaboration with Indian Institute of Rice Research. So far it has been cultivated in 9 lakh hectares in different states, and currently it is being cultivated in about 2 to 3 lakh hectares. And because of this variety, the farmers have got benefit of approx 1500 crores. Okay, so it is helping farmers in increasing their income. And we have licensed the variety to 3 companies. And for this work, we have received 2 prestigious awards. Okay, another important feature this rice has, it is a low GI, low glycemic index rice. Okay, and this was tested by National Institute of Nutrition, which is located in Hyderabad. So, okay, what is GI or glycemic index? So, glycemic index is a ranking of carbohydrates on a scale from 0 to 100 according to the extent of which they raise blood sugar or glucose levels after eating. Okay, so it is ranking from 0 to 100, and if GI value is less than 55, then it is considered as low GI, If GI value is in the range of 55 to 69, then it is considered as medium GI. And if GI value is more than 70, then it is considered as high GI. So when we eat high GI foods, it is rapidly digested, and because of that, there will be increase in blood glucose level. And if we eat low GI foods, it will be slowly digested, and there will be slow increase in the blood glucose level. Okay, you can see in this graph, if we eat high GI food, the blood glucose level will increase very fast, and if we eat low GI food, the blood glucose level will increase slowly. So what next after improved Samba Masuri? So it is improved Samba Masuri is resistant for bacterial leaf blight disease. Okay, but rice has many other diseases. There are fungal diseases. Then there are several insect pests. So we need to add value, or we need to fortify for those additional biotic stresses, also to abiotic stresses, and also to increase the yield. Okay, and what we are doing, okay, so this is one another representative example of another disease in rice. This is called sheath blight disease, and this is a fungal disease. Okay, the bacterial blight is a bacterial disease that is caused by bacteria, but this disease is called by fungus. Okay, and the pathogen is Rhizoctonia solani. You can see the browning on the leaf, or this dead tissues on the rice plant. So these are disease symptoms of sheath blight disease of rice. So like that, there are many other diseases in rice. So what we are doing at the moment is we have developed improved Samba Masuri rice, which is a bacterial blight disease resistant, and also a diabetic friendly rice. So now through marker assisted selection, we are again bringing genes related to many other diseases like blast disease, which is a fungal disease, insect pest like brown plant hopper, then abiotic stresses like drought, lodging, submergence, this is a flooding situation, and salinity, like in near to coastal region because of sea water, there is this stress, salinity stress, and also for high yield. So now we are trying to make a rice crop that has disease resistance for multiple disease. Okay, not only bacterial blight, it should be resistant to blast disease, it should be resistant to sheath blight disease, it should be resistant to insect pest, then it should also should grow in less water, or if there is flooding, then also it should grow, and also it should give higher yield. So at the moment we are trying to develop this variety. Okay, thank you. So hope it was clear.
Thank you, Hiti. Thank you, Dr. Hiti.
Yeah.
So, Mada, are you here? Maybe we can start with few questions.
With the quiz?
No, not the quiz. Maybe for the interaction.
Yeah. So, do we want the students to ask the questions?
Yes, yes. We can give a chance to the students.
Yes, yes.
So, students, I understand that there are a lot of new terms that you have learned during this lecture. I'm sure you might be having many doubts, so feel free to come forward and ask questions to the scientists. I mean, you can, please don't hesitate to ask your queries. If there's something that you didn't understand, is there any concept? Sir spoke about many new things that you might not have studied in your textbooks earlier. So this is your chance to interact and know more about those concepts. I request the teachers to encourage the students, because as teachers, you would also know the standard of your students. So if there's any topic that you think sir can stress again or explain again, maybe you can also share that. You're all currently on mute. If you can raise your hands, anybody who would like to interact, I can unmute them.
Ravani, do you want to pick some of the questions that students had shared earlier?
Uh, yeah, okay, we are getting slowly questions.
Okay.
Is there, so the first question is from PSWR JC Jafargarh. Is there any effect to another crop by sheet blight disease? Any other crop which is affected by this blight bacteria?
No, this is specific to rice, but no, there is a model plant like Arabidopsis, so it affects Arabidopsis also. Okay.
Okay, from PSWR's Jagadgiri Gupta school, they want to know what are the other ways that the plant fights with bacteria.
Okay, so in one of the slide I mentioned that plant induced defense responses. So like then it 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 produced metabolites, secondary metabolites, phenolic compounds, antimicrobial compounds. So that is one way. Then there are defense hormones, jasmonic acid, salicylic acid, and also defense related genes are induced. There is like deposition of callus and also hypersensitive response. These are the ways by which plant induce defense against the bacterial pathogen.
There's an interesting question. They want to know if we can transfer a gene from animals or humans into plants.
Yes, so through genetic engineering it is possible. Like I mentioned, the transgenesis. So using that approach, we can take gene from any source, even from animal also.
Okay, someone is asking if the insertion of blight resistance or any transgene does it affect the crop yield?
Okay, there is a 50% reduction in yield because of bacterial blight disease, approx 50% yield reduction. Okay.
Okay, this question is interesting. How does a plant recognize a foreign body and resist it? It's from Pranav's biology. T-Series JP number. He wants to know how does a plant recognize foreign bodies or bacteria that's invading?
Okay, so even bacterial surface has specific molecules, okay, like exopolysaccharide, lipopolysaccharide. So plant has specific receptors to sense those molecules. So if there is a receptor to sense a specific foreign molecule, then only it can sense and induce defense response. And there are receptors present on the cell surface of plant to recognize those molecules.
Someone wants to know if the viruses can affect any plant organelle in particular. Any plant organelles that are affected by the virus.
So virus attack is also like it hijacks the cell. So it is inside the cell that it hijacks its, say, nucleus. It will go incorporate its genome, integrate its genome into the plant genome, and that's how it multiplies.
Okay, so and there is another question. What are the effective chemicals available to control the bacterial blight disease? And how do we know in a field that our plant is affected by blight?
Okay, so in one of the slide I showed, right, that whole plant, whole field looks like dried out. So in the beginning, there will be patches of field where you can see the plants are dead, and those are symptom of like bacterial blight disease, and then slowly it spreads, and ultimately the whole field looks like dried out. So based on that dryness or dry uh, leaf, you can identify that there is a bacterial disease. Also, this disease symptom we can see on mid vein. If you see the rice leaf, there is a vein in the middle. Okay, so we see yellowing in the this mid vein. That is the disease symptom. Now coming to effective pesticide or how to control the disease. So for bacterial blight disease, there are few antibiotics like kanamycin, but then it's not cost effective. Uh, the price is very high, and also not very effective because bacteria are inside the leaf. When they are inside the leaf and we spray outside, it should reach to the bacteria to have effect, right? The antibiotic should reach to the bacteria. So that's why we don't see a like 100% control even if we use antibiotic. And the best way is to develop resistant rice varieties.
Thank you. And we have one more question from Ogir on the glycemic index. They want to know more about what is glycemic index and what its relation with diabetic patients. How is it helpful?
Okay, so like I mentioned, the definition of glycemic index is when we actually, if a person is given 50 gram of glucose, okay, and the blood glucose level, whatever will be, that is considered as 100, the glycemic index value of 100, equivalent to 50 gram or 100 gram of glucose intake. So compared to that, when we eat equivalent of rice, okay, or any other food item, and then we monitor the glucose level over 2 hours period. So what is difference compared to glucose? That is a control. The blood glucose level of food item. That's how we calculate the GI value. And like I mentioned, that it is on a scale of 0 to 100. So for glucose, if GI value is 100, for rice generally it varies for different rice varieties, say from 50 to 80 or so. Okay, so depending on which rice variety we are eating. And as I mentioned in the presentation, if we eat high GI food, then there will be sudden increase in blood glucose level. If there is sudden increase in blood glucose level, it will affect our organs like kidney, and also if there is sudden increase in blood glucose level, there will be uh, situation. If we eat slow digestive food, then we'll not put up weight. That is also one of the benefit of eating low GI food. Hope
Okay.
I'm clear.
And they've also asked, are there any long-term effects of using or consuming genetically modified crops in humans?
Okay, so one thing here is at the moment any food crop, there are no genetic engineered food crop that are released in India. Okay, so whatever we are eating, they are not genetically modified. Actually, they are developed through breeding only. Only genetic engineered plant that is released in India is BT cotton. So no, not any food crop. And before release, there are several level, like multiple level of testing for safety, food safety, for toxicity, and if it is found safe, then only it is released.
Okay, so I think we are getting similar questions.
Okay.
Mostly they're repeated for the ones we answered during the talk. So if there are no new questions, maybe we can all thank Dr. Hussain for his time and for his lecture. Thank you so much, sir, for coming on board with Milo CCMB project and interacting with our students and telling them more about the Lyme disease and the work that you do. And hopefully, you'll see few of them in your lab later during the internship to learn more
Yeah, yeah.
about the work. Yeah.
Sure,
So,
sure.
yeah. Thank you so much, sir, for joining.
Yeah, thanks, and thanks to Somdatta for this interaction meeting.
Thank you very much, Hiten, for joining us. There are a few more questions which are coming up. I can see in the chat box, though.
Okay, okay,
So
no problem.
probably I will.
Yeah, you want to maybe take them, or
Vinny, what should we do?
Uh, it's
or
okay.
it's a.
But I saw the mostly repeated questions, so I thought we can continue. Did you see any new questions? Maybe we can take it up. Okay, you should go.
I guess it's okay. And maybe people can also write to Hiten
Yeah, yeah.
on his email
So
if they want to discuss it in more details. Also
yeah,
at
yeah.
CCMB, we have a program called as Shadow a Scientist where you can come. And also Milo CCMB will also offer you an opportunity. Before I get into any of those, we have a short quiz like we do after the scientist interaction. So let me share the link here quickly.
Yeah. So, Mata, before we start the quiz, I'll also share the Google form.
Oh, okay.
Yeah. The Google form. So I request all the teachers to fill this Google form so that you can we can give you the participation certificates. Yes. So then you want to see how students have followed the understood the lecture.
It
You
will be
want
a
to
quiz, right?
yeah, it's a quiz. It's
Okay.
fun. Maybe you
Okay.
can stay. It's only for 5 minutes.
No problem. Yeah.
Yeah, you can congratulate the participant, the winner. They'll feel happy. Yeah, Somdata, you please start share the link.
I should? Okay.
Yeah.
All right. So I'm sharing the link in the chat box, everyone. There's a Mentimeter link. Click on it.
Maybe you can share your screen so that
Yeah.
we'll see how many are joining.
Yep.
I'm an audience today. I'll be audience because I'm very poor in botany. Botany and plant biology. I'm an animal biologist. I can see few students, few schools where the student is sitting with headphones properly. Only one student. Manish from Shamir page. Hi, Manish. Say hi. Just wave at your screen. And yeah. And from Shamirpet boys also I see one more student. Is this one boy sitting with the screen. Nice.
About 34 people have joined.
But we have 94, right? So let
Yes.
us also join.
Everyone, you'll find the link in the chat box. Click on it.
I
And
hope
come
by now
join the
you're
quiz.
used to the Mentimeter, how it works and all. I don't think there are any doubts. You just have to click on the link and join. From the schools where there are group of students sitting, you already have a team to answer the questions. Your own team. So even one of your team member knows you the answer, you'll win. But don't waste time and discussions because you have to give the answer fastest to win.
Yeah, faster. If you answer faster, you get more points. All right, maybe I start, Pavani. I see it.
Yeah, 45 is a good number. Maybe we can start.
Okay. First question is up. Say true or false. Microbes can cause diseases to plants. Is it true or false? Please ignore my typo on this screen. Yes, it's true. We have been talking about diseases to rice plants due to microbes. Very good, all of you who got this correct. Let's go to our second question. And for that, we see who's leading right now. Bhavani is leading. Bongir is in second position. Oh, Bongir and Bhavani both are on first. Okay, sorry. Let's go to the second question. True or false again? Some plants can fight microbes better because of their genes. Remember what are genes? Dr. Patel also talked about them a little briefly today. So do some plants fight microbes better because of their genes? Yes, they can. Very good. Okay, Bhavani has kept her position. Question number 3. True or false again? Genes from one plant cannot be transferred to other plants of the same species. Is this true or false? Of course, it is false. We have been talking about getting genes from one plant to another plant via many techniques through transgenesis, through marker-assisted breeding techniques today, right? So genes can be transferred from one plant to another, especially so if they're from the same species. Let's see who's leading the show now. All right, Bhavani is still at position 1, but Jagadish has really caught up. Let's go to question number 4. True or false again? Pesticides can cure all plant diseases. Is this true or false? It is false. Just like the bacterial blight infection that Dr. Patel talked about today, that cannot be cured by pesticides. And that's why we need to look for other methods to make better crop varieties. So not every time pesticides are useful. Let's see who's leading. Looks like Jagdish has got to position 1. Let's go to our last question of the day. Which of these help in making plants of our choice? How will you make plants of your choice? By selecting plants with the correct genetic makeup, by using molecular tools to select correct parent plants, pollination between the desired parent plants, or all of them? It is all of them. So each of these things are important and you can use them to make plants of your choice. Very good. So let's see the final scores. Ooh, KS.
Okay, who is KS? Maybe they can raise their hand. So KS is the winner.
KS is the winner.
Okay, who is KS? Last time also we were not able to locate the parts. I request whoever is with the username or the screen name KS, please raise your hand so that we'll know which school you're from. Okay, I see someone has raised. Oh, Gadwal. Well, unmute. Yeah, I'm unmuted. You can speak. TSWR is Gadwal. Okay, Karunashri has also raised their hand. Who is KS?
Karunashri is KS probably.
Oh, yeah. Okay. Okay. Karunashri,
Many
madam.
congratulations.
Ma'am, can we see you? Where are you? Just a minute. Okay, there is a lot of questions. Karunashri, ma'am. Somdata, can you see the screen?
No.
Good afternoon, madam.
Yes, now we can.
Good afternoon, ma'am. Hello. So you are a science teacher from Jaffragad?
Yes, ma'am, from Jaffargarh.
Congratulations, ma'am. Which standard do you teach?
Thank you, ma'am. Thank you so much. Ma'am?
Which standard? Which class students do you teach?
5th, 7th, and 8th standard, madam.
7th and 8th. Nice. So you have your students with you who joined today? Did your students
Yeah, madam.
Milo CCMB students meet on
Yes, they joined.
They joined?
Madam. They joined, madam. They are in session.
Okay.
Yes, they are in session, madam.
Answers పిల్లలు చెప్పారా మీవా ఆన్సర్స్
నావే మేడం పిల్లలు సపరేట్ గా ప్రొజెక్టర్ లో చేస్తున్నారు నేను సపరేట్ గా మొబైల్ లో చేస్తున్నాను
ఓకే సో ఆర్ ద చిల్డ్రన్ ఎంజాయింగ్
ఆల్సో డూయింగ్ దే ఆర్ ఆల్సో డూయింగ్ మేడం
nice thank you ma'am so
we should also probably congratulate Jagdish. For the first time, it's a student who has scored so well. And I also
where is
see
Jagdish?
From class 9, T Spiris,
Jagdish, raise your hand. I don't see Jagdish in the username. Can you please raise your hand, Jagdish? From whichever, okay. Gadwal? Is Jagdish from Gadwal?
It says Tirumalaipalam.
Okay, is this Jagdish from Gadwal? Tirumalapalam. Yeah, Tirumalapalam. Jagdish, if you're there, you can unmute ma. Tirumalapalam. Try unmuting. You can talk to Somdata, ma'am.
Hi, Jagdish. We cannot figure out where are you in this screen.
I unmuted him. Can you enable your video, ma'am? Jagdish, can you enable your video? Yeah, I think he has enabled. Can you unmute? Yeah, he's here. JC Boys with his science teacher.
We're at Gadwa.
Jagdish,
yes. Now I see you. Hi, Jagdish. Many congratulations.
Thank you, ma'am.
For the first time in our quiz, it's a non-teacher. It's a student who has scored so well. And I also see that your options came a little late, and that's why probably your score went a little down. Very good.
Thank you, ma'am. Thank you, ma'am.
All right. So now let me get to a little more serious business. So all these times after the scientist interaction, we did quiz, but those were quiz for just having fun, for you to summarize, for you to remember the main points that we discussed in the interaction session. But now in April, we will do a serious quiz. In this serious quiz, every school here is expected to send us nominations of students. So we are going to have 3 quiz, and from each school, I would like to see 1 student under each theme. So basically 3 students from each school. The 3 themes are going to be genes and diseases, microbes and infections, nature and conservation. So there are 3 themes, and there will be 1 student for each theme from each school who can participate in the quiz. And the quiz questions will be based on the videos that you have seen so far and the scientist interaction sessions. Now, each of these videos and the scientist interaction sessions are on YouTube. So Dr. Pavani will be sharing those links with the teachers, and the teachers are expected to share those with your students so that they can go through those, revise whatever they have been discussing and listening to so far. And the question paper will be based on that. The quiz will happen on Mentimeter again. So which means that all the kids who are participating in this quiz will need to have access to either a mobile phone or a laptop or a computer with internet. And you have been using Mentimeter now for 3 times at least. So I'm hoping that all of you are comfortable with how Mentimeter is to be used. So the test will happen in middle of April after the school exams are over. So we haven't decided on that date yet, but Pavani will be in correspondence with all the teachers here, and we will come up with a date that is convenient for all of you. So we'll take care of that. But the teachers will have to ensure that you send us your nominations in, by 10th of April probably, so that we have enough time to work around everything. And we will send you the date of the test. And then in this test, under each theme, 10 top students will be selected. And these 10 into 3, so 3 themes, right? So these top 30 students will be spending a week in CCMB. So people who will be selected in the genes and diseases quiz will be spending time in labs which work on those. So for example, you heard Dr. Tangaraj, you heard Dr. Karthik Bharadwaj. So when you come to CCMB, you will be going to their labs. People who will be selected for microbes and infections will probably be going to Manjula Reddy's lab and Hiten Patel's lab, because they work on microbes and infections. People who will be selected for nature and conservation will be going to the Laboratory for Conservation of Endangered Species, which is a specialized laboratory for nature conservation. So, and each one of you will be, not each one of you, like the top 10 students who will be selected in the quiz for these themes will be getting a chance to spend 5 days, which is basically 1 working week, to be with us and see how scientists work and how what kinds of questions they're addressing in their labs. All right. So this is what I have to tell you today. Another point that I should remind the teachers: when you're nominating your students, please keep these 3 things in your mind. We would really like Pavani and I had a discussion over this, and we think that you should select your students on these 3 points. One is their motivation and their ability to learn complex topics, because it might be a little beyond what the students have done so far. So we need to see that motivation in the student when they're coming to CCMB, and their ability to understand English and communicate well enough in English with the scientists, because most of the scientists at CCMB will be either speaking in English or in Hindi. There might be scientists who are not able to speak in Telugu.
Thank you so much, Somdata, for explaining. Maybe I'll go through it again in Telugu so that it's clear for the teachers.
Yeah.
So Dr. Somdata Cheppinattu, we'll be taking nominations from each school. 3 nominations from each school. 3 under 3 different themes. Okay, student for DNA and genes, the second student for nature and conservation, third student under microbes and diseases. Okay, so 3 students teachers nominate change. And when you're nominating, It's not mandatory or essential that the student has to be a topper in the class. If you find any student who is motivated, interested to participate in the project, and who you think can understand the research, అక్కడ scientists చెప్పే రీసెర్చ్ కానీ వాళ్ళు ఎక్స్ప్లెయిన్ చేసే టాపిక్స్ కానీ పిల్లడు లేకపోతే ఆ పాప అర్థం చేసుకోగలరు అనుకుంటే you can nominate. And as Somdatta was mentioning, most of the people in CCMB, both scholars and scientists, they'll be non-Telugu speakers, so who can communicate in English, they'll have an advantage. Easy ga ardham avutundi. And I'm not expecting like posh vocabulary and all that, but basic communicative English. వాళ్ళు చెప్పేది వీళ్ళకి అర్థమైతే చాలు, వీళ్ళు అడగాలనుకుంది అర్థమైతే చాలు. ఓకేనా? సో వైల్ నామినేటింగ్, ప్లీజ్ consider these criteria. So we want 3 nominations from each school. Each select చేసిన students అందరికి, let's say if we have 230 schools, all 230 schools నుంచి the students will be participating in the quiz. So ఏ థీమ్ లో నామినేట్ అయిన స్టూడెంట్ అదే థీమ్ క్విజ్ పార్టిసిపేట్ చేస్తారు. వాళ్ళు జీన్స్ మైక్రోబ్స్ క్విజ్, రైట్, మైక్రోబ్స్ లో నామినేట్ అయితే మైక్రోబ్స్ క్విజ్ ఉంటుంది వాళ్ళకి. సో యూ కెన్ ఆస్క్ ది స్టూడెంట్ టు ప్రిపేర్ ఓన్లీ ఆన్ దోస్ టాపిక్స్, మైక్రోబ్స్ అండ్ డిసీజెస్. ఓకే, ఆ సైంటిస్ట్ ఇంట్రాక్షన్స్, ఆ వీడియోస్ చూస్తే సరిపోతుంది ఆ స్టూడెంట్. సో ఇట్లా ప్రతి థీమ్ లో క్విజ్ తీసుకున్న తర్వాత విల్ బి సెలెక్టింగ్ టాప్ 10, and this quiz will be happening on Mentimeter again, just like the ones that we did now. So ఈ టాప్ 10 స్టూడెంట్స్ కి 1 week internship, 1 week అంటే సాటర్డే సండే తీసేసి మండే టు ఫ్రైడే 5 days internship ఉంటుంది CCMB లో, where they'll be visiting the labs who are working on their selected topics. మైక్రోబ్స్ మీద వర్క్ చేసే వాళ్ళు, నేచర్ అండ్ కాన్జర్వేషన్ మీద వర్క్ చేసే వాళ్ళు, they'll be visiting those scientist labs to learn more about the experiments that they do and the research that they do. ఓకే, సో ఈ ఇంటర్న్షిప్ ఎప్పుడు ఉంటుందంటే మీ ఎగ్జామ్స్ అయిపోయిన తర్వాత ఉంటుంది. ఓకే, మీ ఫైనల్ ఎగ్జామ్స్ అయిపోయిన తర్వాత సమ్మర్ హాలిడేస్ లో మీకు ఇంటర్న్షిప్ ఉంటుంది. ఓకే, సో 2 teams, 3 themes అన్నాం కదా, దాంట్లో 2 themes CCMBకి వెళ్తారు ఇంటర్న్షిప్ కి. ఒక థీమ్ నేచర్ అండ్ కన్జర్వేషన్ లో సెలెక్ట్ అయిన వాళ్ళు they'll be going to LabOnes, laboratory for conservation of endangered species, LabOnes కి వెళ్తారు. ఓకే, any doubts for students or teachers in this? Okay, so it's a great opportunity. Malli 30 students launch kuda will be selecting few students to participate in the next programs. So that we can select few students from them for the Young Innovators program as well, kada?
Absolutely.
Yeah, so we have a very wonderful program at CCMB where you can come up with your own project ideas, and the CCMB team will mentor you on developing the project. So దాంట్లో కూడా ఈ 30 students వాళ్ళకి ఛాన్స్ వస్తుంది. And you're asking to send the concepts and video links. The videos are already available on the Milo CCMB channel, the animated videos. Just you click Milo CCMB on YouTube, you'll get all the animated videos. The live recordings of the scientist interactions are all in the T-Series YouTube channel, for which also I'll be selecting a playlist link soon. I'll be sharing that link. And one more thing, ఈ మీరు సెలెక్ట్ చేసేటప్పుడు 8th to 12th స్టాండర్డ్ ఏ స్టూడెంట్ అయినా మాకు ఓకే, బట్ కన్సిడరింగ్ 10th వాళ్ళకి ఎగ్జామ్స్ ఉంటాయి కాబట్టి 10th వాళ్ళు రారు, 12th +2 వాళ్ళకి కూడా ఎగ్జామ్ ఉంటుంది కాబట్టి వాళ్ళు రారు. సో 8th, 9th, 11th ఎవరైనా ఓకే. ఓకేనా? ఇఫ్ యూ థింక్ ద 8th standard student is very interested and he can understand, he or she can understand, you can send him as well, or you can send the 11th standard student. So 8th, 9th and 11th standard, but not 10th and 12th, as they'll be having examinations, finals. ఇంకేమన్నా డౌట్స్ ఉన్నాయా? ఈ డేట్స్ అవన్నీ కూడా will communicate it in the WhatsApp group. Exactly when we'll give you good time. As Somdatta mentioned, the quiz will be somewhere in the 2nd week of April. That too once you finish your exams. So don't worry, focus on your exams as of now. When you have some free time, you can go through the videos and lectures to follow the topics. And also I advise, when teachers are nominating, please consider the students' interest as well. వాళ్ళకి 3 themes లో which theme they are most interested in, please consider their interest as well, so that they'll enjoy it more.
So people are asking how to get nominated for the exam.
Teacher will nominate.
Yeah.
Teacher can nominate. I'll share a Google form where you can enter the nominated students' details, so that I'll have a database of how many students are actually participating. Any student who did not attend Milo CCMB, can we select them? Okay, so that's a very good
I
question.
mean, you have the right to select, but then if they have not attended any of the Milo CCMB sessions, the chances that they will be winning the quiz is a little small, in my opinion. But then you are the teacher, you will take the decision.
Themes once again. Can
Yeah,
you repeat
Jean,
it?
I am writing in the chat box. How many students from each school? There are 3 themes. I have put 1 student under each theme. So 3 students, but under 3 different themes.
I hope it's clear for everyone. Given a doubt from there, teachers, you can share it in the chat box now, or you can also always send me a WhatsApp message. I can answer your doubts. So
The dates will be told to you. Anumula boys wanted to know. But it
share
will
the
happen
dates.
after the exams.
Yes, in April after your final exams.
All right. So we will be meeting again soon for the big quiz. So everybody do well, and excited to see you all soon.
Yes. Thank you so much. Thank you everyone for the participation. Thank you all students. And we hope to see few of you later for the internship program. Thank you. All the best for the upcoming quiz. Keep learning. Bye. Bye everyone. Bye, Sundata.